Okay, I think we can go ahead and get started. Welcome back, everybody. Thank you for staying with us today. So our next speaker is Perez-Riverol Perez. We are very, very excited to hear from Yasset and learn more about developments for quantitative mass spectrometry-based proteomics in PRIDE. So Yasset holds a PhD in biochemistry and an undergraduate degree in software engineering from Havana University. And currently, Yasset is a technical lead at the European Bioinformatics Institute, so at EMBO EBI, where he leads a variety of efforts related to proteomics team. In particular, he leads the PRIDE efforts, PRIDE archive, PRIDE cluster, and proteogenomic resources. So we are very, very happy to hear from Yasset. Thank you so much for joining us.
Let me check. Do you hear me?
Yes, we can.
Okay,
now we are paying attention to the audio.
Okay.
Expect some strange screen sharing from me if something goes wrong.
Okay. First of all, thank you to Olga for inviting PRIDE to present our recent effort on proteomics reanalysis and also data archival. I will be talking about PRIDE archive, which is our main mission. Then I will try to touch a little bit about SDRF, which is one of the efforts we recently did for trying to facilitate to our community the reanalysis of public proteomics data. And then really actually a recent effort from PRIDE team to, with other teams like OpenMS, to try to reanalyze large-scale public proteomics data. As Nuno mentioned, PRIDE is also a member and actually one of the founders of Proteome Exchange. Proteome Exchange is a consortium that groups multiple databases, including PRIDE, Massive, JPOS, and IPROX, Peptide and Panorama. Magisha will be talking after me about Panorama. And I think the main, a part of storing data in each specific archive, I think the main functionality, I would say, is the implementation of standards and guidelines to for data submission. I think this is one of the main tasks of Proteome Exchange. That means that if someone tries to submit data to PRIDE or to Massive, they would be following the same guidelines. Then PRIDE mission actually, in particular, has two main missions. The first one is to try to archive all type of public proteomics data. Try to also make this data available for others. And at the same time, and this is our second mission statement, is to provide evidence from this public proteomic data into other resources. And actually, I would say biological and knowledge-based resources like UniProt, Ensemble, and Expression Atlas. The whole idea is that making actually the data that is not only for mass spectrometries and also people who knows mass spec, but also other biologists who come, or clinicians that come from a different perspective to knowledge-based databases like UniProt, Ensemble, and Expression Atlas. They can find out actually public mass spec evidence, expression data, peptides, PTMs, and so on. PRIDE is a huge ecosystem. We actually try to highlight the major components of PRIDE. Take this to here. The major components to PRIDE in our recent paper in January this year. And it all started with the submission of the data set. We have two main kind of submissions called partial and complete. The user used actually one of our submission tool to submit the data to PRIDE. Then it arrived and multiple pylons actually are validate the data, submit the data, notify the user that the data is actually in our hands. Also that give some credentials for them. Reviewer of the data set that to be used under the review process. then the data remains private until the user asks Pride to make it public. The main, actually, and I will basically highlight this, the main difference between a complete submission and a partial submission is that in a complete submission, the result files are based on a standard file format. And actually, Pride for a long time, over the years, has spent a long time with software tools, bioinformatics tools like Mascot, MaxQuant recently, and others. I mean, in Protein Discover, not only open source and free tool, but also commercial tool to actually export their results into a standard file format like MCTAB, MCIDML, MCML, and so on and so forth. And what is actually the relevance of this is that whoever does their analysis and then export and actually share their data in a standard file format, anyone can understand that those results. Then, and there is some features in Pride that actually use heavily those complete submissions. Then we have some internal pipelines that actually convert this data into, for complete submission, into peptide evidence and actually give to multiple resources, especially to UniProt and Ensemble, give actually evidence at the protein level of which actually particular peptide or PTM has been seen in a particular data set, and there is a link from those actually databases into to Pride. Then the data set as it was submitted originally by the user with their own FDR and so on and so forth, and we'll talk a little bit about that, actually goes into a knowledge-based database, and someone who, for example, is interested in one particular PTM actually can go, and is actually looking into UniProt, actually have the link to the original data set and the original submitter of the data that was used to actually find that evidence. This is really important for us. It's actually giving credit to the users that make their data public. And this is related with all this archival process and also providing the evidence into other databases. We also provide databases to other systems like OmicsDI and biosamples and so on. And we try to make as visible as possible that people can actually find the data from Pride. The data sets in Protein Exchange has been growing, like I would say, like no any other community in recent years. I mean, the adoptions of public, the position of public data in proteomics is actually, I would say, it's quite common that any paper right now nowadays has their own Protein Exchange accession. And actually, we see this in the statistics. I mean, you can see here how still we are incrementing. You know, the number of data set is actually incrementing like 12% per year. But actually, we see actually almost 500 data sets per month are submitting to a public domain. And I really would like to point out this number. This means that every month, in average, until 2021, 500 data sets were actually made public within the proteomics community. And Pride actually stored like 80% of the data in Protein Exchange, and this is why it's so relevant for us. And we'll talk a little bit about what means for us this 500 data set per month. The number of instruments, I mean, the data in Price is kind of, I really would like to highlight this. It's coming from multiple internet, really a variety of instruments, not from any single provider. We don't see any major provider, only of some increment of some technology. And still the number of data sets, as I said, is public. And it actually, this is really interesting, is how well how Europe, US, and also Asia actually is well represented in the number of submission to Pride. It's also really important to know that you don't have only human data in Pride and in the public domain. You have, of course, modern organisms are well or more represented than other organisms, but actually there is multiple data set that go from normal, I would say, a human data set into metaproteomics data set and other species, plant proteomics, and so on and so forth. How do you search it? I will go a little bit about how do you search in Pride. Pride has enormous functionalities for searching data. If you go to Pride Archive, it's our main repository to search for data. You will be able to search data set using different keywords. And actually our data, our system will provide you with some highlights about keywords that you can actually use for searching your data set. You can sort here data set by publication, the publication date, relevance, and so on. When actually, there is a one, let me go back because this is really important. I think you will see this batch here for each data set in the right corner of each data set. And this is actually how relevant the data set means, how much a data set has been reused by others, how many times someone has seen this data set, how many times someone has downloaded this data set. And this is actually relevant for us because when the data is growing, it's actually important for us to give to the user that is searching for relevant data functionalities that allow them to search for the right data set. And the number of downloads, the number of views, the number of citations that the data set gives some sort of impact, can be used as a proxy to understand how relevant for the community is a particular data set. A data set description of Pride, if someone has a right to Pride, look like this. But I would like to highlight a new feature that we also have in Pride, that using metadata, we actually suggest to the user new similar studies. And this is relevant for us because, as I said, as the number of data sets is growing, it's actually really important for the user to have some highlight about what similar data set to the one that I'm looking right now. Actually, I can try to go and check how this data set look like. Then when the data is deposited in Pride, we use the complete submission because they are in a standard file format. We can read them. They are in MCML, MCML, MCTAB. We can read those lists of peptides and proteins. And actually, we have some QC on top of that. For example, following the HPP guidelines, we control the number of amino acids and so on. Also, we try to compute an FDR for the complete data set. And all these lists of peptides are stored in one database. We call this Pride Peptidome. And this is mainly the relevant, let's say, high quality peptide that has been identified and reported to Pride, but our users, the user that deposit the data in Pride. And this is actually like 2,000 projects. I mean, it's like 10% of Pride data, and it goes over almost 2 million of peptides and almost 1 billion of 1 million for unique peptides. And I mean, it covers huge amount of species. Important thing is that the user can go and search right now in Pride Peptidome for one particular peptide, and they can see for this particular peptide how many times has been seen, in which project, from what species, in which protein has been seen, and what is the spectra that is related to this project. Also, which disease and tissues this peptide actually has been found or identified related with the metadata of the project. But as I said, and I think I pointed out this multiple times, this is related. This is the data that has been submitted to the user. If someone reanalyze the data, analyze the data, for example, with, I don't know, MAX1 exported into MCTAB, then the MCTAB that we can read, then we throw this data into this system, and then we store all those peptides into Pride Peptidome that user can actually check where this peptide with this PTM has been seen, which projects, okay? Associated with which tissue, with disease, and so on. And finally, you can check one particular peptide and actually see the spectrum. What particular spectrum is actually the best evidence for this particular peptide, something similar to peptide habits, but not as a result of the reanalysis, but actually as a result of a high quality peptide coming from the submissions of the user. This data actually is connected to UniProt, as I said. And then if you go to UniProt, you can say for one particular peptide, what is actually the protein, for one particular protein, for one particular sequence, if there is any evidence in UniProt. This is a working process right now, and still is in depth, but soon will be there as other resources like Peptide Atlas and Proteomics DB. But something that is actually in production is we also map those peptides into genome coordinates, and then we throw them into, we actually enable the user to see those peptide evidence through track hubs. A track hubs is a genome mechanism or resource that you can actually look for data set and then visualize the track hub in a genomic context. And this is really relevant because we are really interested on connecting the genomic with the proteomics. Then if you go to a region of the genome, probably this is not the best example, but if you go to a region of the genome and a particular gene, you want to see where this sequence of the genome has been identified. If you have modification, also see the modifications, and then link to the particular evidence in PRI. Then this is what PRI does with the original data submitted to PRI. We store that data, we actually curate with some pipelines and try to extract high quality evidence from submitted data set and push those evidence as it was reported by the user and their pipelines into UniProt and Ensemble. However, PRI, as Massive and others, is actually really interested on not only have identification results like peptide, PTMs, and so on, but also work in the quant part. I mean, really being able to pass into those resources quantitative information. We actually have some sort of a statistic. I think this, I really thank Juan Antonio Vizcaino, the team leader of the team, who has done an incredible work trying to figure it out how the data in PRI is actually downloaded and used in different manuscripts. And we see this is actually how the data has been increasingly also downloaded from PRI. But actually, if you see this word cloud, you can actually see that mainly has been reused for meta-analysis, proteogenomics, verification of PTM, data integration, isoform, and benchmarking, and spectral library generation, but not for quant. Then we went actually over what is actually the main reason, and we have a really nice paper in 2016, more or less, when we actually highlight what kind of things, or we spot that were actually the problems that triggered that people is not using data for quantification reanalysis. And the main problem is that within PSI and protein exchange, the information around the sample and the relation to the file was missing. And what that means is that we collect a lot of metadata around the project level, but because there is a lot of experimental designs, there is a lot of variables under study, protein exchange in general and UPO-PSI never managed to capture really well the experimental design, how samples were connected to data files, especially instrument data files. This actually means that every user of the data set needs to download the data set, understand the paper, and actually get in contact sometimes with the author of the paper and perform the quantitative reanalysis. And this is for automatic reanalysis and large-scale reanalysis of data. This was actually a big issue because you cannot build pipelines unless you have someone reviewing or annotating those data sets in a way or in a configuration file for something like MAX1 and so on. Then last year, actually, we lead a community within UPO-PSI. Nuno mentioned this effort to have something called a sample to data file format that captured not only the sample metadata, but also metadata around the file and actually the experimental design. Where my sample is, in which particular file actually my sample is, and what actually how is the relation between that file in terms of, for example, if one sample has been fractionated, then one sample is into multiple files, then the relation is one to many and things like that. Then the file format the main feature of that is actually extremely simple for users and submitters of pride data, meaning that you can, in an Excel file, write your SDRF and submit it with the submission for others to reuse in the future. It is ontology-based, it's compatible with other existing PSI file format, and it's focused on reuse. And I will put an example about that. I have one question here. What is the difference between a spectral library, background, proteome, and peptide list? Okay, I think a spectral library, we generate from the peptidome the spectral library, but in the peptidome, the aggregation happens at the peptide level. I mean, from all the peptides that we have in the evidence that we have across all the data set, we put some quality metrics there to actually assess the quality of the peptide and then pass that peptide list into UniProt and so on. We also generate the spectral library, but the spectral library has the problem that you need to aggregate everything, and it's time consuming in terms of scalability. Then you cannot do, because you need to take all the data set and take all the spectra, cluster it together, and then get the peptide for each particular cluster. We do that to generate the spectral library, but these are two independent processes. We do this for the spectral library, and the peptide list is passed to the peptide to UniProt and Ensemble when actually it's assessed the quality at the peptide level. And the background protein, I think probably Nuno knows more the concept than me. Then going back to the presentation, SDRF looked like this, and this is actually really relevant. I mean, you have the sample here, you have, for example, the characteristic of the sample, for example, which disease, and some information about the phenotype. For example, you can be studying cancer, which is the disease, but actually the phenotype is that you have different conditions for the same sample. You can be having some samples that are normal and some samples that basically are necrotic tissue and so on. You can put as many as possible, as many as you can, information about the sample itself. We have, if I have multiple presentations, you can go to actually the paper, and we explain we have templates to require more data, more metadata for some experiment than others, and so on. Then there is a really important part, which is actually if this is a biological replicate or this is a technical replicate. There is also here between the relation of the file and the sample, if this is a technical replicate. And actually, if this is a fractionating experiment, I need to know if this row is fraction 1, fraction 2, fraction 3, and so on and so forth. And why this fraction order is important, because some tools, like for example MStats or MaxQuant, they actually use match between ROMs and this kind of algorithms. I guess you heard about that during the course. But basically, you try to find features from one biological replicate into another for the same fraction. Then for that, you need the whole experimental design to be accurate, because if not, you can be looking into a different fraction when you're actually doing the match between them. But finally, there is something called the factor value, which is actually the variable under study. And the factor value defines, for example, what is the technical replicate or the biological replicate. In this case, factor value is phenotype. And then this is a clear labor-free experiment. In this case, no fractionation, but we have plenty of examples around TMT, SciLab, iTrack, and other experimental design. In this particular presentation, you can see how my experimental design is translating into an SDRF, okay? Then when you submit a data set to PRIDE, now you provide all the raw data, all the result files, and actually the SDRF with the sample to data relationship. This go into PRIDE archive. And finally, for some data set, the ones that has the SDRF, you can see in PRIDE the metadata for each particular data set, all the files. And actually, we throw this information also into other resources like Biosample. Then if someone is looking here for one particular sample, let's say necrotic tissue in adenocarcinoma in human proteomics data, in Biosample, they can find a data set and a sample for it, and actually the connection to another data set, for example, transcriptomina, and so on. We have reannotated around 200 data sets. PRIDE team itself, actually, we have a repository in GitHub when we reannotate data of other people to, for our for our own reanalysis, but also for others to benefit out of this effort of reannotating. You can go to our resource, to the GitHub, annotate your own data set, the data set that has been already deposited, and then we actually pull that to Pry and then make available for others. Okay, then we actually think we have solved in some way, but this is a standard file format that is actually going, still is working towards. We think that we now have a well-filed format that can be adopted to reanalysis for public reanalysis and also reproducible reanalysis. If someone goes see the NSDRF, you can have your own pilot and then analyze the data and then push back to Pride. Okay, you can put the reanalysis back to Pride. You can do a submission, and then you just need to define what is that section that you actually base on this particular section. But Pride also does reanalysis. Pride actually reanalyze data to push quantitative data into other resources at EVI. Then in 2019, 2020, 22, between 2019, I think we started this, we did some sort of try to find out what were the best tools are out there, or tools that were more used for reanalysis of data, to put those tools into a Pride environment to reanalyze data. And we did some voting pools and so on with users in the community. Also, some research about the number of data sets, the number of tools in the public domain, the ones that are free and so on. MaxQuant, as all of us know more or less, and Skyline still are the predominant tools. And you can see here how other tools like Masco, for example, were dominant for a while, but now MaxQuant have taken over them since the first release of MaxQuant. And actually, you can see really well that MaxQuant and Andromeda hasn't changed the adoption over the last 2 years. It is really relevant. Skyline has been growing and making more popular. PixDB, Comet and MSGF Plus as a search engine, they keep active, people still citing them. And as we all know, FragPy and Alexei has been doing incredible work during the last 2 years, and MSFrag has jumped into the scenario, and then people is using that. Then I have always we in Pride, and the best tool is there is a plethora of tools. The best tool is the one that works for you. Not all, you really need to know your tools. You really need to know you have experience on your tool. And actually, we know, for example, that some teams use still search engines and tools that are not really, let's say, popular or not widely used, and still they get a lot of results with that and so on. Then we know what is the trend, but we really want to solve other issues within public reanalysis of data. And that's why we actually, since there is one problem for reanalysis that we know, we started reanalysis data with MaxQuant. But one of the problems that we have actually is that with MaxQuant and other, let's say, monolithic tools, you need to have all the data pushing this tool in this monolithic tool and then do the reanalysis. And this is actually great because those tools scale, MaxQuant scale quite well right now. But the problem is that if you are trying to reanalyze 500 data sets per month, as I said before, even if you don't aim that, you aim 20, 30, whatever, you are not really using the capacity that you can have in a cluster. You cannot disseminate all the data across compute, distributed, and then get back the results, okay? Because there is no tools and not so many. TPP has this approach, but not so many that can actually do that. Then to reanalyze data with really large scale, we really need something that is reproducible, that you can run now and in a year time, and actually you get the same results. That can be stream oriented, meaning that you can actually launch a specific part of the data, get the results. But if you actually have a problem with this particular step, you can actually launch again that particular step and then finalize your results. It should be focused on parallelization, distribution of the task, as I said, reproducible before. Then Last year, we did a review of things it was published in Proteomics, of tools that can be used for public reanalysis of data. And we really want, we drafted something like what kind of approach should be following or in the future proteomics to approach more other topics like, or other fields like transcriptomics, is in a way, we really need to split functionalities from compute and actually from the tools. How do you deploy the tools? This, you can read this review. We did this for proteomics and metabolomics. In a way, we want to have something that you can split small steps into compute steps, into different nodes. We really want something that the user doesn't need to, every time you go to a computer, let's say you are in your personal computer, need to install here, but then you go to AWS, which is something that you can do for reanalysis, and then install everything there. Then you go to HPC in your local university and then install everything there, which takes a long time. And sometimes when you install, the version that you used to have is not available anymore. And this creates a lot of reproducibility issues. And then since a year ago, more or less, we have been working something called QuantMS, which is a cloud-based reanalysis proteomics infrastructure and pipeline. It allows you to perform TMT, labor-free DDA and DIA-LFQ analysis on the cloud, based on the public proteomic data, but also the standard file format that we have available. And I will go quickly over this. This is actually the flow chart of QuantMS. It doesn't develop any new tool. It uses existing tools from OpenMS tool suite and other developed wrappers to actually grab a specific steps of a reanalysis or a proteomics analysis, and then grab them and then connect them over file format, especially standard file format, to have a final result. I will go quickly over this. This starts with a raw file or MCML. If the raw file is provided, we have a thermo raw file parser, which is a tool that we developed a couple of years ago. We transformed this into MCML. The user can switch between MSDF Plus or Comet, or use the combination of both search engine and combine the results using the consensus ID from OpenMS, and then perform percolate on top of that. And finally, if it's LFQ, it will do protein LFQ. It will do the quantitative analysis of LFQ. And if it's actually TMT, then it will go through isobaric detection, and then protein inferring and protein quantification. And really important, at the very end, you will have NCTAB file format. I always said this. We are basing everything in standard file format, which basically means that you can do a complete submission back to PRI, and other standard file format for downstream analysis like MSTAB or Trickler. If you want to do DIA, we use DNN for in silico library. It's library-free analysis. DNN has been parallelized with the help of Badin, the developer of DNN. And actually, you will have at the very end also MCTAB and MSTAT for DNN. I will go also from independent step of this pipeline. The pipeline, as I say, the identification part between TMT and LFQ is shared. You can use any of the search engine, Common or MSGF Plus. And then what is different is actually how you perform the protein inference, because in the protein inference part, also in the quantitation part, we do match between runs to get the final results. In TMT, we use a different strategy for comparing to get the protein quantification different in using the channels and the TMT channels. Then let me move this here. And this is really important. The starting point of this pipeline is an SDRF. Then if we have deployed this in AWS, then if I have an SDRF available, okay, and I have a protein database, you will see one example, then I can pull the data from PRI, directly from PRI. And in the SDRF, there is some parameters already encapsulated in the SDRF. You can perform the reanalysis, the identification, the quantification analysis, and then push into SDRF, into an S3 bucket, the final result. And the final results are in MCTAB for the protein, peptides, and PSM. You can see here, for example, MCTAB, how it looked like. An MCTAB is a tab delimited format that has the protein section with the quant values, the peptide section with the quant values, and the PSM information, which PSM, which spectrum was used to identify this particular peptide. How do you run it? I mean, you just need to download something called Nextflow, which is the workflow system that is used to distribute the task, control which task is failing, resume when one task is failing, resume the pipeline for that particular task. And for example, you can do really amazing things like, I will respond to that question now, but you can do amazing things like, for example, imagine that you are doing the peptide identification with Comet, and Comet is consuming too much memory for this particular raw file. If it failed due to memory issues, the pipeline automatically, Nextflow, actually try to allocate a memory with a higher, actually, memory run to run the data set. But not only that, it can be, for example, you have an issue with a CPU, you have an issue with the specific file, and then the pipeline has a logic and the workflow has a logic to control these things. Yeah, I will provide a list of a website to, I will provide the list to QuantMS, where you can find the information about the data set, and sorry, the workflow, the modules that it runs, and so on and so forth. Then, in particular, this is really amazing. I mean, this is, I would say this is an amazing technology. When you run, it's a command line thing, workflow system, but also you have a web that you can connect to the workflow. But behind the scene, what is happening actually is that you have a workflow, and then you have one input SDRF with actually the experimental design. What is the database that I'm trying to search? In this case, will be, for example, I think this is UniProt and a combination of UniProt and Ecolab, I think, for this particular data set. You can add decoys, and there is a lot of parameters. You can attach the decoy database. You can decoy proteins to the database. You can define the search engine. You can say, I want to search with MSDF Plus or with Comet, or I want to search with both, and then you can define the thread. But the amazing thing with this workflow is actually that you can run it in your own personal computer with Docker containers. You don't need to install anything. You just have Nextflow and Docker, and then it will pull every tool needed, MSDF Plus, it will pull Percolator, it will pull for you OpenMS tools, and run everything in your local machine. If you have a different version, it will run the different version for you, because we keep track in biocontainers of every single version for each particular tool. And this is really important, because that means that if someone submitted data set to Pride, a reanalysis to Pride, and they say, I have used this version of this particular Nextflow QuantMS workflow, we can actually reproduce all the tools that were used at this particular moment, okay? And this is amazing, because anyone can reproduce the same analysis at any given point. The second thing is that you can use in multiple infrastructure, not only your local computer, HPC, Google Cloud, AWS, any of those architectures are now supported. Let me move quickly. This is actually how a workflow looks like. And actually, I put this particular example when you actually run, for example, the DIA label-free data analysis. For this particular generation of the in silico library from the database, actually, we failed the first time in one node because of memory. And automatically, the pipeline will find out, okay, I don't have enough memory for this particular task. Let's try to allocate in the cluster if there is a node available to run this. And then the step is successful, and you can continue the analysis. In Nextflow actually provide amazing possibilities, and QuantMS, of course, as one of their pipelines, to, for example, know and actually trace your pipeline. For example, in this particular case, for this LFQ pipeline, I know now that, for example, Comet is actually allocating more CPU than MSDF Plus. But for example, in terms of memory, we know that, but it's really good to know to have a report of that shows actually this to the user. This actually help you to, for example, if you are reanalyzing data, as we are doing, you can actually here decide, okay, because MSEF Plus is consuming so much memory and I don't have enough big memory nodes, then I will run all of my analysis. Even if I lose PSMs and peptide evidence, I will run all of my analysis with Comet, which actually do not almost consume memory. And actually, you are trying to actually use actually nodes with less memory. QuantMS, and this is a development we have done. We are really interested in doing QC. You will find out that at the very end, we have a QC report for your experiment, where you can do QC at the multiple level. At the raw file, you can see intensities, for example, multiple variables, charge states, for example, across all the MCML you have submitted. I would I would give this URL, and you can basically check which kind of reports we give them. And for example, if you're doing really nice at the peptide level and at the final reports, if you're doing, for example, a multi search, you can see here that MSEF Plus slightly overperform always Comet at the PSM level when we perform the analysis. All the report will give you this information. It will give you number of PSM for MSEF Plus, a number of PSM for Comet, and the final peptides that were identified, and some really nice plots. We have a lot of plots, for example, number of peptides per protein, distribution of precursor charges for identify and no identify. I want to be quick. Then at the very end, when you finalize your experiment with all your configuration, you will have a folder like this, where you actually even capture intermediate files. I mean, we actually store this configurable. I mean, people can decide to do not store an intermediate file. Let's say if you just want to have the MCTA with the final PSM, it's fine. But also you can store, for example, the PSM for report for each specific search that you perform, MSEF with MSEF or Comet. And of course, we are also completely aligned with MStats. We're using MStats heavily. At the very end, we use some default configuration for MStat in the LFQ pipeline and also in the TMT pipeline, and you can actually get the MStat report as one of the output of the data set. I think we are now developing some particular training application using MStat that can actually pull, because we really know that the downstream analysis really is not really a static thing. I mean, users like to, define the parameters themselves and actually explore particular proteins and so on and so forth. Some examples. Last year we published in bioinformatics the first use of the pipeline to detect actually non-canonical peptides and push these non-canonical peptides into Ensemble. We actually analyzed more 6 data sets in price, 65 cancer cell lines, more than 20 million MS, and all this data was pushed actually into Ensemble. And this is a better example how this looks in Ensemble. Now, an example for one specific gene, you actually can see PTMs, or you can actually see variants if you actually have a variant in your peptide for this particular experiment in prime. All those data sets, if they are analyzed right now with MaxQuant, or in the future with QuantMS, will go to Expression Atlas to visualize the final expression values. Expression Atlas is a resource at EVI that has more than 10 years. It's mainly focused on RNA expression data. But since 2019, we have been adding more data set. Right now, have more than almost 80 data set in proteomics. And you can see actually the results like this. You can actually check one particular reanalysis if it's one particular data set, and you can see the proteins that are expressed for this particular data set in one particular tissue. And the amazing thing is that you can see this for one particular gene, you can see this for transcriptomic and proteomic in the same resource. Then Pride will be continually analyzing data and pushing this data into a spreadsheet atlas for a more multi-omics view of the proteome and the transcriptome. And I want to also highlight that recently, spreadsheet atlas also have single cell transcriptomic data, which means that you can see what we call bulk RNA data, proteomics data, single cell, everything within the same resource. Thank you so much. I mean, this is quite a lot of information. I tried to capture what we have been doing recently in Pride. Thanks to OpenMS team for working actually with us in the QuantMS pipeline. Vadim also has been working actively with us to be able to reanalyze DI data. And of course, Pride team, especially to Juan Antonio this time, which is the team lead of Pride.
Thank you so very much. Yes, this is really, really interesting. And I think that this is an amazing contribution to the community. So there are several questions in the Q&A, but in the interest of time, I propose that you answer them in writing, if it's okay,
yes,
so that we have enough time for our last speaker. But thank you so much. And I'm really, of course, very excited to see that the MS Stats is being useful in this effort. So this is really, really wonderful work. Thank you. Thank you so much. So last, but certainly not least, we have a presentation which focuses on yet another repository. This repository is Panorama. Panorama is focusing around the Skyline ecosystem and is one of the main repositories that works specifically with quantitative experiments. And I am really, really happy that Vagisha joins us at the May Institute for the first